To illustrate the relevance of analytical and bioanalytical chemistry in the development of biopharmaceuticals, I've selected two case studies. The first one is about a monoclonal antibody that is used for the treatment of certain types of breast cancer. As we've already seen, a monoclonal antibody is a large molecule of about 150,000 Dalton, so there's a lot of possibilities for modifications. The monoclonal antibody in question is against the HER2-Neu subunit of the EGF receptor. By blocking the signaling through this subunit of the receptor, breast cancer cells can be prevented from dividing and multiplying. This is only true for HER2-Neu positive breast cancers, which account for about 25% of all breast cancers. Suffices to say that this antibody trastuzumab has recently been also been licensed as an antibody-drug conjugate, where the antibody delivers a toxic load to the cancer cell. However, this antibody-drug conjugate will not be treated in this case study yet. Herceptin, or trastuzumab for injection, has been licensed for the treatment of metastatic breast cancer with overexpression of the HER2 receptor kinase. It is an interesting example of a combination of a bioanalytical assay, a diagnostic assay, to confirm the overexpression of the HER2 receptor kinase, and the subsequent treatment with Herceptin. This is what is known as a companion diagnostic. So first you stratify the patients with a diagnostic test. In this case it's an immunohistochemistry test. And then you treat the patients that respond to the treatment with the monoclonal antibody with significant success. I believe that this will be more and more the standard way of treatment in the future, also called personalized treatment. The heterogeneity of antibodies and also of Herceptin can have multiple reasons, including deamidation, changes in the glycosylation pattern, and C-terminal cleavage. We will see how, and this is already an example from some 10 years ago, how analytical and bioanalytical methods can shed more light on the heterogeneity of an antibody. If you would like to see more details, there's a very nice paper by Harris and co-workers from Genentech where this antibody was developed, detailing how they worked and discovered and identified the different variants of this antibody leading to charge heterogeneity. The first analysis was performed with strong cation exchange HPLC. Let's quickly rehearse the functional groups of ion exchange materials. We have strong and weak ion exchangers. Strong ion exchangers means that they are permanently charged, no matter what the pH. Let's say, no matter what the pH across a reasonable range applicable to proteins. Weak anion exchangers change their charge state with the pH, so the pH can be used for elution as well. Here we see strong anion and weak anion exchangers, where the stationary phase is positively charged. At the bottom we see strong cation and weak cation exchangers, where the stationary phase is negatively charged. As said before, for Herceptin we used a strong cation exchanger with sulfonic acid groups on the stationary phase. This ion exchanger is negatively charged across the whole entire pH range from 1 to 14. If one looks at Herceptin in this way, one discovers multiple forms. A major form 3, somewhat intermediate forms 1 and 4, and some minor forms A, B, 2 and C. All these forms must differ in their charge state, otherwise they would not be separated by a strong cation exchanger. Going back to the principle of strong cation exchange, we assume that form 3 has more positive or less negative charges than form 1. We now need additional analytical methods based on mass spectrometry to find out where the differences are and also later whether these differences may be detrimental to the biological activity of Herceptin. As discussed earlier, isoelectric focusing in a polyacrylamide gel is another powerful method to look at charge heterogeneity. Remember, the isoelectric point of a protein is determined by the number of positively charged and negatively charged groups in a protein, which are equal at the isoelectric point. If we look at peak or form 1, then we can see that its isoelectric point is more negative than that of peak 3. It looks as if peak 3 has one negative charge more or one positive charge less. The difference in charge state seems to be 1. Please notice the exquisite resolution of the isoelectric focusing gel. Even in a protein of 150,000 Dalton, this gel can resolve single charge differences. You can also see that peak 1, which was collected after strong cation exchange, and peak 3, which was also collected, are in themselves still not homogeneous. They contain at least 4 forms themselves. If we look at peak 4, it seems again to be a bit more basic than peak 3, explaining its later elution. However, this difference is only gradual, and there is a significant overlap between the forms in peak 3 and peak 4. This is easily explainable by the fact that the separation is not complete on the HPLC, but there may be other reasons as well. I did not note all the other forms, but the principle remains. It is interesting to note that peak 3 and peak 4 can be partially separated by strong cation exchanger. Remember that the mechanism on a strong cation exchanger is not purely based on charge, but that also hydrophobic interactions can play a role, depending on the backbone of the stationary phase. To gain further insight into what is different between these forms and where the modifications are located, we need to prepare a peptide map. That means we need to digest the protein in defined ways, using proteases. I've already mentioned that before. This is a, let's say, model protein where I would just like to remind you the process of peptide mapping. First, a protein is digested with a protease that has specific cleavage sites like trypsin. Trypsin cleaves C-terminal to lysine and arginine unless these amino acids are followed by proline. In most cases, we also like to cleave the disulfide bonds and to alkylate the SH groups as mentioned for the in-gel digestion. This prevents that peptides remain bound to each other, which makes analysis difficult, and it also prevents that disulfide bonds are formed again after disulfide bond cleavage. Such a strategy was applied to Herceptin to gain further insight into its modifications. Let's take a look at the 2 major forms of Herceptin, peak 3, the major peak, and peak 1, the second most abundant form. Chromatogram A at the bottom shows peak 3 and the top peak 1. It's not difficult to spot the difference. Peak named N30 has diminished and a new peak D30 occurred in form 1. So here we can already see where the modification is. But how did people arrive to localize this modification? This was done by mass spectrometry, and in this case by MALDI-TOF mass spectrometry. The molecular weight of these peptides was measured and shown to be 1 Dalton different. D30 was 1 Dalton heavier than N30. So not only did form 1 have less positive charges or more negative charges than form 3, but it also gained 1 Dalton in mass. It is now the question, what is the chemical or biochemical reason for this instability or heterogeneity of Herceptin? To this end, we need to go back to the chemistry of the amino acids, and notably the amino acid asparagine and aspartic acid. It's been known for quite a while that asparagine can be converted to aspartic acid under certain conditions by a chemical reaction called deamidation. What does this imply? If you look on the left side, asparagine has an amide group in the side chain. This makes it a neutral side chain and gives it a pI of 5.41. Aspartic acid, the deamidated form, which is also a natural amino acid, has a carboxylic acid group in the side chain with a pK of 3.65, giving the overall amino acid a pI of 2.77. That means it is significantly more acidic than asparagine. From the point of view of a strong cation exchanger, that means an additional negative charge would be introduced into the protein by a transfer from asparagine to aspartic acid. This would, or could at least, diminish the affinity for the negatively charged stationary phase, explaining the earlier retention time. You can also see that the mass of aspartic acid is about 1 Dalton more than that of asparagine, since NH3 is removed and replaced by a water molecule. This is shown here in this reaction scheme. See again asparagine on the left side with the amide group. This can be hydrolyzed by water under acidic or basic conditions and dependent on the sequence context to give aspartic acid. So you add a water molecule of 18 Dalton, but you lose an ammonia molecule of 17 Dalton, which overall makes that aspartic acid is 1 Dalton heavier than asparagine. As mentioned earlier, an antibody of the IgG family is made out of a light chain and a heavy chain. All in all, it has about 650 amino acids. And this times 2, because there are 2 light chains and 2 heavy chains in an antibody. The boxes here show parts of the antigen binding regions. These are the binding sites that are certainly important for biological activity. If you look into these binding sites, you see particularly position 30. We see an asparagine threonine sequence. Threonine is a small amino acid and thus facilitates the deamidation of asparagine to aspartic acid. As you will see, this reaction goes through a cyclic intermediate, and this requires quite some flexibility in the chain. This is facilitated by small amino acids. As this asparagine 30 is in the binding site of the antibody, we might expect some effect on the activity of this protein, so it is an important site to keep in mind. There are other sites in Herceptin that are critical, for example the site here, asparagine glycine. Glycine is also a very small amino acid, and thus also here deamidation could occur. I will show a little bit later on that we found this deamidation does occur not only in vitro but also in vivo in patients, likely inactivating Herceptin during the course of the treatment. As mentioned already, the deamidation reaction proceeds via a cyclic intermediate, a so-called imide peptide. Let's look at an asparagine peptide here that is deamidating. The first step in the reaction is a nucleophilic attack of this peptide nitrogen onto the carbonyl carbon of the side chain amide bond, forming a cyclic imide, also often called a succin imide. Now this cyclic imide can open in 2 directions. By the hydrolysis with water, either to the natural L-aspartic acid peptide or to the unnatural L-isoaspartic acid peptide. Note here that the peptide chain continues on the side chain of the amino acid, so essentially the peptide chain has jumped from the main chain to the side chain. This leads to considerable structural perturbations of the monoclonal antibody and often, and other proteins as well, if it happens, and often leads to inactivation of the corresponding protein. Let's take a look and see whether we can discriminate between these two possibilities analytically. The colleagues at Genentech did a considerable amount of work to bring this all into perspective and to look at all the different forms. This is, however, necessary if you want to bring a biopharmaceutical product to the market. As mentioned earlier, biopharmaceuticals are by nature heterogeneous, and it is important for approval to identify and to explain each and every heterogeneity that can be observed, and more importantly, to show whether or not this heterogeneity has an effect on biological activity. You may imagine that this is a considerable task. That is why analytical data form the major part of any registration dossier of a new pharmaceutical product. But let's go back to our different peaks. Peak 3 was the major form. It covers 73.8% of the peak area, and it is as it's expected from the DNA sequence: asparagine in position 30 of the light chain, asparagine and asparagine in position 55, the other position I pointed out, in the heavy chain, and aspartic acid in the heavy chain at position 102. If we look at form 1, which constitutes or represents 13.6%, then we see, as is already pointed out, it's deamidated on one of the two light chains from an asparagine to aspartic acid. So these two forms together make up about 87% of the total protein. Let's look at form 4, which represents 8.5%. This is an interesting modification since all the asparagines are still intact, but one of the aspartic acids, in position 102, has isomerized to isoaspartic acid. This can happen just as the deamidation reaction can happen via the cyclic intermediate. So aspartic acid, the same nucleophilic attack can happen forming the 5-membered ring imide, and this one can open in both directions. Often the isoaspartic acid form is more stable and thus will accumulate over time. I don't want to go into all the individual forms. You can read more on this in the paper given below. Just one thing is interesting to see. Form C, for example, has the succinimide in position 102. This is actually the intermediate prior to the generation of the isoaspartic acid. So actually, in the heterogeneity of this antibody, you see part of the chemical reaction mechanism happening from aspartic acid to isoaspartic acid. You can appreciate we are looking only at a very small part of this monoclonal antibody, and thus you might appreciate how complex the situation is if you look at a protein of 150,000 Dalton in such detail. Actually, if you look back at the previous table and the previous slide, you will see that there is another form with isoaspartic acid. Not only form 4, which has isoaspartic acid at position 102 of the heavy chain, but there's also form B, a very minor form, which has isoaspartic acid at position 55. This is the reaction product of the deamidation of asparagine to aspartic acid in one of the binding sites of the heavy chain, though this might also be an important modification. Here is an overview over all the modifications that we've looked at. Asparagine 30 can deamidate to aspartic acid 30. Asparagine 55 can generate aspartic acid 55 or isoaspartic acid 55. And aspartic acid 102 may isomerize to isoaspartic acid 102. It's interesting to note that all three modifications are in binding sites of either the light chain or the heavy chain. This is not so surprising, as the binding sites of antibodies are usually somewhat disordered loops with considerable structural flexibility to adapt to the surface of the antigen and bind it tightly. Structural flexibility is an important prerequisite for deamidation, as the 5-membered intermediate imide ring must be formed, and this is very difficult in structurally restricted areas such as beta-pleated sheets or alpha helices. Recent work from my group shows that this is not just an in vitro phenomenon of protein stability or instability, but this happens also in vivo during the treatment of breast cancer patients. This was a collaboration with PRA Health Sciences and the Oncology Department of the University Medical Center here in Groningen. You see an example of one breast cancer patient that is treated for more than 1 year with trastuzumab or Herceptin. We have followed with liquid chromatography tandem mass spectrometry the different forms at Asparagine 55. The original form, remember, has an asparagine in position 55. However, during the course of the treatment, this asparagine is deamidated and isomerized to isoaspartic acid to a considerable extent, maybe something like 15 to 20%, and some of it deamidates to aspartic acid 55. We choose another peptide that is stable to determine the total amount of Herceptin. This is given here in peptide area units. Translated into concentrations, it's in the microgram per milliliter range, but this is not most relevant. It can be assumed that the isoaspartic acid form is probably inactive, although this still needs to be confirmed. Most interestingly is that different patients show very different profiles of isomerization, deamidation and isomerization, and thus inactivation of Herceptin. So this must be taken into account during the treatment and to establish the proper dosing regime. These data could not be obtained with previous analytical methods, so this is the first time that such a complex molecule is analyzed in blood of patients and that specific modifications due to in vivo metabolism are shown that potentially affect the activity of the biotherapeutics. This in vivo drug metabolism for biotherapeutics will probably be a growing field, as one has to understand why different patients need quite considerably different dosages of very expensive biopharmaceutical products. It's now important to understand whether the observed modifications have an effect on biological activity. This was tested in a cell antiproliferation assay of a breast cancer cell line called BT474. Without going into all the details, you can see that peak 3, which is the main form with the expected sequence, has an activity relative to the mixture, which was set at 100%, at 141%. Peak 1 has somewhat diminished activity, corresponding to about the average of this heterogeneous preparation, 98% activity. Peak 4, which has an isoaspartic acid at position 102, has almost completely lost its activity. Remember, this was about 8.5% of the preparation. It is thus very important to monitor the stability at this position, as batches of monoclonal antibody may be completely inactivated if position 102 goes from an aspartic acid to an isoaspartic acid. What happens at position 55 is currently under investigation. We expect that it will also have an effect on activity, as asparagine 55 is in direct contact with the HER2/Neu receptor kinase. However, these data are not yet in. We are currently pursuing these together with the colleagues from the Oncology Department. Let's summarize briefly our findings. Importantly, it is shown that the monoclonal antibody Herceptin is not a homogeneous product, as we are used to from chemically synthesized drug molecules. However, this is not unusual for biotherapeutic proteins, which are pretty much all heterogeneous products. It is thus important to analyze them batch by batch and to assure that this heterogeneity is constant within certain limits, as the chemical heterogeneity of the product is linked to its biological activity. It was shown that deamidation of asparagine at position 30 occurs in the light chain, and it affects the biological activity, although only to a minor extent. Other positions have also been shown to be either deamidated or isomerized. For example, aspartic acid at position 102 of the heavy chain was isomerized to isoaspartic acid, and this led to an almost complete loss of anti-proliferative activity. Considering that Herceptin and other monoclonal antibodies have many more sites that may potentially be modified, a review by Beck et al. Showed what is all possible and what has been observed in Herceptin, where we looked at the deamidations in position 30, 55 and 102, or the isomerization here. There are other possibilities, for example the transformation of glutamic acid to pyroglutamic acid. There can be oxidations of methionine, for example, with +16. There can be possibilities or changes in the glycosylation of Herceptin, and many of them have been shown. And there can be proteolytic cleavage at the end of the heavy chain, removing one lysine residue, to name just a few. So it is a formidable task, a formidable analytical task, to characterize such a large molecule as Herceptin and to assure that it's produced in consistent quality. Analytical and bioanalytical chemistry are thus instrumental to the current success of biotherapeutic proteins. Without the advance in mass spectrometry and liquid chromatography, this would have been unthinkable. Registration dossiers at the FDA or the EMA contain mainly analytical data, so this shows how important analytical and bioanalytical methods are for the current day modern biopharmaceuticals. Let's look at case number 2, a case that I worked on during my time as biotechnology researcher in Strasbourg, France at Transgene SA. The protein in question is called Hirudin. It was first isolated from the blood leech Hirudin medicinalis. You can see a bit more about this fairly interesting medicinal animal on this YouTube film. Preventing coagulation is the speciality of the blood leech, as it feeds from human blood or from animal blood. Thus it must prevent blood from coagulating while it is feeding. To this end it injects anticoagulant molecules into the bloodstream. This was the basis for our investigation to isolate the active compounds from the blood leech, which turned out to be a peptide of about 65 amino acids, to clone it and then to express it recombinantly in yeast and to produce it on a larger scale. Actually, Hirudin is on the market as a pharmaceutical product nowadays, primarily to prevent venous thrombosis and as an alternative to heparin. As mentioned, Hirudin is a high-affinity specific thrombin inhibitor that was originally isolated from the blood leech. It has an affinity in the femtomolar range. It is thus the so far highest affinity thrombin inhibitor known. Hirudin, under the name lepirudin, was produced in the yeast Saccharomyces cerevisiae and licensed as an anticoagulant to avoid blood clotting in thrombocytopenia, for example induced by heparin. In work published in 1993 already in biochemistry, we looked at the deamidation of Hirudin and also the succinimide formation, so this is somewhat reminiscent of the monoclonal antibody. If you want to know more about this work and the early days of liquid chromatography mass spectrometry, you can read the publication here in Biochemistry. When moving towards clinical phase, we had to characterize an impurity in Hirudin. As you can see, this was a very minor product of less than 1%. You can imagine how high the main peak of this chromatogram is going. This is actually still a classical chromatogram with a UV absorbance detector. In order to characterize this impurity, we had to isolate it first, and as you can see, it's not very well separated, so this was a major task requiring lots of starting material. However, after major efforts, we were able to purify the compounds in the impurity, and it turned out that the impurity was composed of two different molecules related to hirudin, impurity A and impurity B. The major product is shown on the top. It had a calculated molecular mass of 6,906.5. It was thus a large peptide, or a small protein if you like. Impurity A and impurity B had about the same molecular mass, which was somewhat 17 Dalton less than the major product. This sounds like a simple analysis to date, but in 1993, or just a couple of years before when we did the work, this was a major breakthrough, as electrospray ionization and MALDI had just been commercialized. Reminiscent of the work with Herceptin, we first cleaved hirudin into smaller products. However, hirudin does not contain many lysines and arginines amenable to tryptic digestion, so we had to resort to another protease, which is called endoprotease Glu-C. As the name indicates, this protease cleaves C-terminal to glutamic acid. The glutamic acids are indicated up there at the numbers. So this provided us with the following products: 1 to 49, 1 to 43, 50 to 61, 44 to 49, 62 to 65, and 1 to 61, as not all cleavages were complete. I indicate a little triangle on these fragments, as these are the modification sites, as we will later see. We found the mass differences in this fragment. That means it must be somewhere between 1 and 43 amino acid, and we found the modification here in the fragment between 50 and 61. So this already allowed us to localize the two different modifications, or should I say the two same modifications, as they led to the same mass difference. We will see. Summarizing, we see here the observed mass differences of these fragments, which we isolated and measured again by electrospray ionization mass spectrometry on, in those days, still fairly experimental analytical systems. Fragment A and fragment B in the impurity A had both a mass difference between 17.5 17.7 about. Fragment C had the mass difference in impurity B of 17.0, while the other fragments corresponded pretty well to its expected mass, as they did over here. So this confirmed that impurity A had a modification of minus 17 Dalton about between amino acids 1 and 43, while fragment C had the same or similar modification between amino acids 50 and 61. Looking again at the reaction mechanism of asparagine deamidation, we should focus on the L-imide, the succinimide intermediate. As you can see, it's formed by an intermolecular nucleophilic attack and with the loss of ammonia. Now ammonia has a mass of 17, so this was a very likely explanation of what we were seeing. We think we were seeing succinimide intermediates at two different places in hirudin, one between amino acids 1 and 43, and one between amino acids 50 and 61. It was surprising, and in those days the first time, that such intermediates were stable enough to be isolated and characterized by mass spectrometry. In parallel, and in collaboration with the group of Robert Huber in Munich, we did crystallize hirudin in the complex with thrombin to better understand its interactions. At the same time, this crystal structure allowed us to localize the asparagines that were deamidating to the succinimides, and again they were found in flexible loops at the outside of the molecule. Here we see asparagine 33 at the tip of this loop, and here we see asparagine 53 again in a fairly mobile region next to asparagine 52, which was interestingly not found to be deamidated. Hirudin has then its N-terminus, which is active and goes into the active site of thrombin, and the C-terminus, which helps in this interaction through ionic interactions. We therefore hoped, from a production point of view, that these deamidations would not have an effect on antithrombin activity. This was actually confirmed. You see, the major component has an inhibition constant of 1.15 picomolar, while impurity A and impurity B have essentially the same inhibition constant. We could therefore safely assume that impurity, which was less than 1%, should not affect the overall biological and hopefully pharmacological activity of Hirudin, which later turned out to be true, as the product is also on the market now. Let's summarize briefly this short case study too. While the thrombin inhibitor Hirudin is less complex than Herceptin, it is still not a homogeneous product, as is customary for biotherapeutic proteins. It was interesting to see that the cyclic succinimide could be isolated and was stable enough to be observed. This was never been done before, or was not known before. Succinimide formation had no effect on thrombin inhibition, which was very important from an application point of view.